My name is Leimkuhler Grimes, and I'm a professor at the University of Delaware. I'm excited to tell you about the probes that my team is developing with support from the NIH Glycoscience Common Fund. So currently, my team is interested in studying bacteria. And if you're watching this video, chances are you are too. Perhaps you're interested in studying the human microbiome, those 3 to 5 pounds of bacteria that are living in or on you right now. Or maybe you're interested in studying a pathogen, such as Mycobacterium tuberculosis. Well, whatever pathogen or bacteria that you're studying, they all share something in common. All bacteria wrap themselves in a jacket of carbohydrates called the peptidoglycan. These jackets are essential for human health, as they make an excellent target for antibiotics, and in addition, we use them as an immune recognition element. The molecular structure of the bacterial peptidoglycan is a composition of a polymer of 2 carbohydrates and a variety of peptides. And the carbohydrates are widely conserved between microorganisms, and so there's 2 of them. N-acetylmuramic acid, shown in blue, and N-acetylglucosamine, shown in green. N-acetylmuramic acid is really specific to the bacterial cell wall, and my team and I thought this would be a great place to put our probes to learn more about bacterial peptidoglycans. The whole focus of my team's project is to develop tools that will be usable by the microbiologist and immunological communities. We hope that these tools will help answer fundamental questions such as what are the structural features of the jacket that set it apart from different organisms? And what are these small immunostimulatory molecules that are constantly being shed from these bacteria? And do they differ between microorganism to microorganism? It's really hard to study these carbohydrates that are in the bacterial peptidoglycan, as you can't see them. So my team and I thought it would be amazing if we could install little flashlights inside the bacterial peptidoglycan so we could begin to visualize it, to learn about the structural features, and potentially use these flashlights as molecular beacons to realize what are the immunostimulatory fragments. This is an actual image generated from my lab and my team. And so now I need to tell you the story of how we generated this. Thankfully, I work with a really amazing group of dedicated scientists. And when we were thinking about installing these probes in the bacterial peptidoglycan, we quickly realized that we would need large volumes of these carbohydrates. And thankfully, Kristen Demister is an excellent synthetic organic chemist. And so Kristen designed synthetic methods so we could rapidly access large volumes of these carbohydrates. So this process that I'm going to show you is now patented, and Kristen can produce these molecules on very large scales. And in them, on these molecules shown in red, this azide and these alkynes, we can use these as probes to install our flashlights. So now we have access to the bacterial cell wall building blocks. And so now we needed to figure a way how to get them onto the surface of the cell. Enter Hai Liang. Hai Liang is an excellent molecular biologist and biochemist. And Hai designed an enzyme delivery service so we could install these probes on the surface of the cell. Hai had to work through 14 unique individual biochemical steps to install these probes on the surface of the cell. And a key step to this was utilizing a page from Christoph Mayer's playbook, who taught us that there's 2 enzymes, AMGK and MUR, which participate in a recycling program to convert Kristen's building blocks to these very useful UDP MURNACs, which then get synthesized or biosynthesized to the surface of the cell. So now with the building blocks and the enzyme delivery service in place, my team and I felt confident that we could potentially remodel the bacterial peptidoglycan. So we turned to E. coli as a model system. And we needed to give E. coli a few things. First, we had to give it those recycling enzymes, AMGK and MUR. So we genetically encoded them into the genomes. And then we needed to give them the Kristen's synthetic building blocks, these azide or alkyne-containing NAMs. And we were going to treat the cells with phosphomycin, a compound that would force the cells to use this recycling program and then incorporate into the biosynthetic steps. And so with this in place, then we would do some, as the probes were installed on the surface of the cell, we would then do some click chemistry to install these fluorophores or these flashlights. And then when we look underneath the microscope, so we treat our cells with either the azide or the alkyne probe. And then when we look underneath the microscope, we can see in a normal light microscope, E. coli looked just fine. So the compounds didn't change the morphology of the cell. But when we turn on the flashlights or the fluorescence microscope, now all of a sudden we see beautiful images of the bacterial peptidoglycan. This is the highest resolution image of the carbohydrates in this bacterial peptidoglycan ever generated. And the really nice thing about this method is that we can apply it to human immune cells, such as macrophages. So here the bacteria are in these images. You can't see them. However, when we turn on the flashlights, you can see them. And the nice thing is you can see not only whole bacteria, but you can also see fragments. So we can almost use these lights as a GPS tracking system to sense these fragments as they make their ways through your human innate immune system and begin to understand them. We've done this in an E. coli model system, and we realized that's just one type of bacteria. There's other types, commensal, pathogenic, gram-positive, gram-negative. And we made a list of bacteria that we're actively trying to label. And the best part about this is that I get to work with two dedicated microbe experts, Nina Salama at the Fred Hutchinson Cancer Center and Sloane Segrest at UMass Amherst. And so Nina and Sloane have shown that they can remodel both H. pylori and Mycobacteria tuberculosis using this methodology. And together, Nina and Sloane and I have been working to have an impact so we can ship this methodology and these compounds to labs around the world and the country. So we've shared these probes and this method with multiple labs already, and we'd really like to share it with you if you're interested in using these probes. So we believe our method is simple. First off, we think about the bacteria that you want to remodel. We'll work with you to determine if you need to include AMGK and MURU, that recycling program, in your bacteria. And then we will give you large volumes of those carbohydrate building blocks that you need to remodel. You'll incubate with your cells. You'll do some click chemistry to install either a fluorophore and an affinity reagent. And then once those are installed, you can then do these downstream imaging applications and potentially mass spec experiments to identify very interesting biological fragments. Now, in order to gain access to these probes, either this azide NAM or this alkyne NAM, you can email us at namprobes@udel.edu. We have a dedicated technician in the lab who will help ship them to you and help you learn how to work with them. And if you want to wait until the summer of 2020, these probes will be available in TCI America's catalog, and you'll be able to purchase them through them. So thank you for taking the time to listen today. Please reach out if you have questions, and I'd like to thank the NIH Glycoscience Fund for support for the development of these probes.